Nalini, you all of you first foremost. Nalini, who is he? Nalini, are you submit?
Yes, I submitted.
Okay. I will see. I will check it here. Why should I? Yes, Namrata, Sachin. Is it not your section? Yeah. Your section is 11. Oh. Nitin Teja, Nitin, Bhargavi, Sri Sai Subrat, respond. Whomever I call, please respond. What is your real problem? Is it a net problem? Or else you are out of station? Shashank? Ankit.
Yes, sir.
Oh, why? What's your problem, Ankit?
Sir, submitting, sir. 1 minute.
Okay.
Oh, thank you, sir.
Satvik, Surya Satvik Verma, Nadimpalli Surya Satvik Verma, Vidya, Sri Vidya, 11056 Sri Vidya. One 11009 Shivadutta.
Yes, sir.
Yeah, you submitted.
Yes, sir.
Don't worry, you submitted.
Okay, thank you.
11027 Akhil Venkata Akhil. Kirti Kiran. This is the people only, I think. How much time you want? 11015 Sanjay Shemakurti VVSSN Sanjay 11015 Sanjay. These 11 people, still the date is not yet submit. It's not good practice. These people are not respond. Only one guy is respond. Okay, you have anybody your best friend? You inform to them. They may join or not today's class. I can't understand. Okay. For the topic, genetic transmutation, translation is over. Next is genetic gene code, genetic codon, the coding, genetic code. Sir did not. Who is he starting something? Sir did not check my Diagram I have submitted. Who is he? I will check it. I will check it, all of you. You don't worry. Beside of your PIN number, you put your name. All of you after this class. Your names is definitely I will remember. Okay. The genetic code. What is the genetic code? The double helix of structure of the DNA, Watson Crick, and their anomalous it's non-stop. They're up to reach to goal like that. They with a lot of effort onto that. They identify the gene, the DNA, the hermetically characteristics is passed on through that one. This double helix structure, the shape and their arrangements of the chromosomes, that particular the DNA and RNA containing of the nucleotides. The arrangements, the arrangement of chromosomes is in cis or in trans way of its nucleotides. Accordingly, you will identify. Yeah, the next is genetic code. The genetic code is nothing but of our genetic in translation. So each and every each and every nucleotide is combined as as each and every three is combined as a triplet. The triplet form of amino acids, amino acid 1, 2, 3, 4, like up to 20 amino acids in biomolecules. The lesson in our unit 2 already we know about. And the amino acids, the important amino acids in 20 amino acids we identified. Yeah, this particular the triplet codons wise it arranges in the DNA containing of nucleotides you know well about. RNA containing of the nucleotides you know about. One is the difference, that is uracil. Yeah, it's a mRNA and a tRNA. The messenger RNA is coded, decoded, and tRNA is generates of this particular each and every triplet is a form as an amino acid. This is the thing already we know about of our genetic transmutation. Okay, the genetic code is in a dictionary that is corresponding that is a sequence of the nucleotides and a sequence of amino acids. Amino acid 1, 2, 3, 4, it's like up to 10. Yeah, the 64 genetic codons is very important one. So these codons, 61 it's an amino acids containing, and the rest of 3 is not yet containing amino acids. That codons is differs. Okay, the genetic codon is a set of rules by which the information is in encoded. It's in encoded. This means decoded, like it's in a genetic, the material of the DNA and RNA, or RNA, DNA or RNA, the sequences. It's translated into the proteins by living cells. Yeah, is one of the scientists. Is in the name of the genetic code is in Georgia Gomo. Georgia Gomo, he mentioned, yeah, the great scientists of the Watson and Crick. And next is in Georgia Grom is postulates that the 3 letters of the code, the 3 letters of this particular nucleotides is converts as in a code that is in a universal the language like. These are the UAUUU, uracil, 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 or UAG, uracil, adenine, and guanine. It's like these 3 letters of the code. It must be employed to the encode to that particular 20 standard amino acids is used for the living cells. It's a building proteins. Yeah, this particular the 20. These 20 is it situated of any 3 letters of the code digits. This triplet coding is in Giorgio Gome identified. Next, you know about the nucleus is in a prokaryotics or eukaryotics, but it's in a prokaryotics in a cytoplasm. The DNA is in open DNA, and in a eukaryotics is a nucleus membrane, the nucleus containing of the DNA containing the chromosomes. This chromosomes is in a lengthy one and a short one of this chromatides. Yeah, there it is in a L and Q is mentioned in the particular centriole 2. It's in the same nucleotides is in one side of this chromosomic pair. In a human, the chromosomic pairs is in it 23 chromosomes, but one is a sexual the chromosome. Yeah, the total 46 chromosomes or 23 pairs. Only one pair is in sexual chromosomes, and the rest of all are is in isosomes. Yeah. Now, this all is decoded. And again, that what a particular double helix ladder of DNA structure. The collection of the codes is called as the genetic code. Yeah, of course, is this in a DNA or RNA. The collection of code is called as the genetic code. For 20 amino acids, there it should be actually 20 codons. Each codon should have 3 nucleotides to import. Yeah, the specificity of each of this amino acid, a specific codon and a specific characteristics. It maintains one nucleotide is 2 nucleotide and 3rd nucleotide, the 4 components and 6 components, total is 64 combinations. The most of it is situated 20 amino acids. In this total 64 is not all are not an amino acids. Out of 64, 61 is in amino acids. Yeah, it's the decoded of this particular the nucleotide. The contains and nucleotides converts of the 3 letters of the coding. Yeah, this is of adenine, thymine, guanine is one of the amino acid. The cytosine, thymine, adenine is another codon, and guanine, and cytosine is another amino acid. This particular 3 base pairs is in a code form. Next is an amino acid. It generates. Yeah, this is all. The genetic code can be expressed as in a simple, the tabular form of 64. It's an entire genetic code is in a UUU. Yeah, all are split from Now the double helix structure of the DNA from one adjacent to another adjacent, their arrangements. We're already seen that one also. Yeah, this particular the amino acids, the shorter forms. Each and every amino acid comes under this particular the codons CUU, CUC, CUA, and CUG. These all are come to is in a one considered as in a one amino acid. That functionalities is differs, but each and every codon is different. All of you remember that. Next is carbon and its type. The codon and its type. The genetic code is in the dictionary consists of in a genetic word is codons. Each codon consists of three bases. This particular three bases is triplets. There are only 64 codons. Already we discussed this one. The 61 codons is code for the 20 amino acids is found in protein, but in a 3 codons which do not code for any amino acid. It's not a encoded amino acids out of 64. Well, three only is like that. But at the same time, the types of the codon is the sense codons, signal codons is in start codons, stop codons, the sense codons. Just like the sensory organs, like the sense codons is a codon that is in a code to amino acid are called as sense codon. The single codon, signal codon, and those codons is code for the signal during the protein synthesis is called signal codon. For example, of AUG, UAA, UAG, and UGA. These are all. There are the two types of signal codons. One is the terminating and initiating. The initiating codon and terminating codons, signal codons. Yeah, the start and stop. The codons is the signal codons. The initiation and termination. Next is in a terminating codons is UA, UAA, UAG, and UGA. These are termination codons, or is in the nonsense codons are often referred to as in amber, uchira, and opal codons. These are in a short form of amber, ochre, and opal codons. These nonsense codons. And initiating codons is in AUG. This is an initiating codon. It codes for the first amino acids in all proteins. Codes in the first amino acids in protein. At starting point, it codes for the methionine. The methionine in eukaryotics. The methionine in eukaryotics is formerly in methionine in a prokaryotics also, but it's in a prokaryotics. The methionine in a cytoplasm. In eukaryotics, this particular the methionine in mitochondria. The mitochondria containing of what it is matrix. Yeah, the mitochondrial matrix. We're already where it is a mitochondria and cytoplasm. We know about in our TCA cycle, tricarboxylic acid cycle, the electron transportation point of view. Watson and no, not Watson, Krebs cycle. In the Krebs cycle, in the glycolysis we are identified. Yeah. Yeah, it's terminating codons and initiating codons is differs. The initiating codons of in prokaryotics in a cytoplasm and eukaryotics in a mitochondria. These are all situated. The anticodon, the base sequence of a transfer RNA, tRNA, which pairs in which codon of RNA is during the translation is called of the anticodon. Yeah, is it before we discussed from 3 to 5 or 5 to 3 the arrangement of this double helix structure. The tRNA is in anticodon loop, is in a Obel position. Yeah, this is the position is in a Obel position. What is a Obel position? We discussed in a last pair of the codons. It is in a guanine with uracil or guanine with cytosine. This is in a Obel position. In a pair of three, in the last pair of this particular three is mentioned as in a Obel position. It sometimes is that, but guanine, one of the nucleotide is same. The guanine is it's in mRNA, messenger RNA too. It converts as in a form of either it combines with uracil or cytosine. This Weibull position, Weibull position, we will find out. But it's in a 3 to 5 or 5 to 3. Yeah. The next is the difference between the codon and anticodon. Yeah. The codon could be present in both DNA and RNA, but it's an anticodons is always in RNAs, in mRNA and the tRNA, and never in DNA. The anticodons never we are find out of the anticodon activity. We are not find out in DNA, but it's always in RNA. The codons is written from 5 to 3. It's a detection where it says in anticodons is usually written as in a 3 to 5 the detection. Yeah, the codon is in 5 to 3, and anticodons is usually 3 to 5. The before slide I think 3 to 5 and 5 to 3. Yeah, the anticodons of some tRNA molecules have the pair with more than one codon. Pair with more than one codon is maintained. The codons are sequentially arrangement of the nucleotide, and the nucleic acid is strand. That particular the strand, while it is in anticodons or in discretely is present in a cell with amino acids is attached or not. Accordingly, you find it, and the codon defines which anticodon should come the next with the amino acids to create the protein strand. The anticodon helps to the bringing a particular amino acids its proper position during the translation. Yeah, this is the case of mRNA and the tRNA translation. The translating way is only we find it. The next, these are the all characteristics is exhibits the features in the genetic codons. Yeah, this in the triplet code is in a Comolos. It's not, I'm not understand. Okay, Comolos, and the next is non-overlapping code, the coding dictionary, and degenerate code, and universality of the code, and non-ambiguous code, and chain initiating code, and chain terminating code. We will find it. These are all the properties we will find out here. Yeah, is in a chain termination codons or in a chain initiating codons. The universality of the codon and non-ambiguous the codon, and the degenerate The degenerate code is in mRNA point. Yeah, we will find out it here. And non-overlapping code, triplet code. The triplet code is major. This is for triplet code. We'll find out this particular genetic triplet code of 64 in the codons. Out of 64, 61 is in amino acids. This is the triplet code of genetic codons. Okay, codons. The universal type. Universality is in a genetic code. Is in universally is in a AUG. Is in a code. Is a codon for methionine in mitochondria. But the same the codon of AUG is a code for isoleucine in cytoplasm. With the same expectation is noted is in a genetic code is in a universally. But where it is, this is the condition. The same AUG is coded for one case is in methionine and another case is in isoleucine. The isoleucine is in a cytoplasm. The cytoplasm is in a prokaryotics. Already we discussed. In mitochondria is in eukaryotics. Yeah. This type of, with some assumptions, is noted to the genetic code in universally. This is some of the assumptions. Some expectations is noted for regarding up to this this AUG and AE. And non-ambiguous, the genetic code is in a non-ambiguous. Is in a one code is can't specify for more than one amino acid. Yeah, one for one only this particular the characteristics wise, and the non-overlappings, no base, they cannot participate in the formation of the more than one codon. Yeah, it means that it's in a code is a non-overlapping code. If it's may overlap, then it's a lot of confusion. It's not the universal codon. Yeah, it's a continuous translation. The gene is in a transcribed by translating continuously from a fixed starting point to the fixed stop point. Stop point and starting points both are then fixed in the punctuations are not present in between the codons. Yeah. The code has is in polarity. The code has in a definite the direction for reading of the message, which is referred to as in a polarity. Yeah, the character of the polarity, it may shows the reading of the message from the left to right or right to the left, from 3 to 5 or 5 to 3, which is specified for the different amino acids. For example, of UUG stands for the leucine, for that the same time is right to left of GUG, which is in a stand for valine. This is the difference where genetic codon polarity we will find out. This is a degeneracy. It's a degeneracy of the genetic code. Degeneracy is in a triplet codon as in a 64. The first nucleotides, yeah, second nucleotide and third nucleotides, the point of view. The nucleotide of the first nucleotide is in a uracil or in a set of cytosine or adenine or the guanine. UCAG. In second nucleotide point, second nucleotide of arrangement of the here all are cytosines. And the next in a second nucleotide, all are adenines. in UAA and UAC, UAA and UA and CAU, CAC and CAA, CAG, AAU, AAC, all are in a second nucleotide is the same. The same sequence of the second nucleotide is in the left hand side. UCU, UCC, UCA, UCG, and CCU, all these are second. But in the first case, the first nucleotides, 4 uracils and 4 cytosines and 4 adenines and 4 guanines wise. It's arranged. The next is in a third nucleotide point. In the third nucleotide point of view, it's called as a degeneracy of genetic code. Is it changes in third nucleotide point of view. Next is in a Wobble hypothesis. What we've seen is before. The Wobble hypothesis, where we are find out is it from 3 to 5 or 5 to 3. In this 3 to 5 or 5 to 3 of this Wobble hypothesis of the identical the leucine of tRNA from the left to right or right to left. What way it's mentioned, you see. In a case of mRNA, and at the same time, in a case of tRNAs, from the left to right or the right to left, we're already identified at here. Yeah, this is what it is. Yeah, mRNA is in one side, it's in a Wobble, and the phase of this. The Crick postulates the Wobble hypothesis is to account for the degeneracy of a genetic code. According to this hypothesis, that the first two bases of codon pair according to the normal base pairing rules, with the last two bases of this anticodons, and the base pairing at the third position of the codon is in Wobble. Yeah, like for guanine is its connection with the uracil, or at the same time the guanine is connects with the cytosine. This is already in that before sites we are identified in a Wobble pairing. So in a Wobble pairing, this is of our Wobble position or Wobble pair. Same guanine in a third pair. The guanine is its shares decoded with mRNA. Yeah, it transmits an uracil, or the same guanine is in the cytosine. This is of our Wobble position. There is a position, the tRNA to mRNA, but here it is in a Wobble pairing of this particular, the guanine to cytosine or the guanine to uracil. This is the thing. The next Wobble hypothesis explained in degeneracy. The Wobble hypothesis explains in a degeneracy of the genetic code, that is in existence of the multiple codes for a single amino acids. In although there are 61 the codons for amino acids and one number of tRNA is for less around is 40, which is due to the obeling codons. The next biological significance of degeneracy of the genetic code. The code is not a degeneracy. The 20 codons would be designated amino acids, and 44 which would be leads to the chain termination. The probability for mutating the chain, mutating the chain in a termination, it would be therefore to much higher with non-degenerate codes. The degeneracy is from non-degenerate codes. It will possible. Yeah, this is of the DNA to RNA. The proteins, say RNA is in mRNA and tRNA is decoded. Critical significances point of view, just only the mutation only up to only you learn it. So need not to be beyond the level of the name. The point mutation, silent mutation, and missense and nonsense and frame shift mutations. These mutations is possible. Then codons will change genetic, entire genetic scenario will change. Yeah, here is enough to these all mutations and the types and everything is no need. It's in a nonsense mutation. Okay, I stop share it here. The next is find it. Whenever mutation takes place, then definitely the codon disorderness, codon disorderness, we will find it. Yeah. Now, once you watch, how to remember is the easiest way for this particular genetic codons in the triplet codon of these amino acids. You see how to remember this genetic codons, this triplet form. These are all in the first letter and the second letter and the third letter point of view. Total 64 triplet codes. What we have read, you see.
So how to remember? As we all know, this genetic code has been discovered in 1961 by Nirenberg and Mathai, along with so many
Nirenberg
names
and
will
Mathai.
come. So in 1961, it has been discovered by Nirenberg and Mathai, Matthaei. Correct? For the cysteine, we shall not use any mnemonics. You can remember by University Grant Commission for the cysteine, UGC, and the first one is the UGU. 3 codons has to be eliminated. Those are stopped, like UGA, UGA here, UAG, and UAA. So as we know, UAA, UAG, and reverse of it, UGA. These 3 are stop codon. So we need not to use any mnemonics for it. So we start from the first. There is a first mnemonic, that is the poor lover lost in middle village. So it is the first easiest mnemonic, P for phenylalanine, first 2, then lover, leucine. The next 4, as we know, genetic codes are degenerated. So 4 and 2, this 6, UGA2,
Yeah, this is the
they all can
way
code
of
for the leucine. So this is the degeneracy of the
mnemonics,
code.
Bandaguttu, mnemonics through. We will. Is the poor lover, the lost in the middle village. Just like of our what it is for domains wise and our classifications, everything we are in mnemonics way. You will remember the first. The stop codons and start codon of UGC, University Grant Commission, you may remember, that is of our UGC. Here, of our UAA and UAG and UGA, except UGC. These are all our stop codons. Yeah, stop templates. And the next is in poor lover, the lost in the middle village. The first 2 of phenylethylamine and the leucine, UAA and UAG. It's a lover, is in a lover of L is in a leucine, and the poor is in a phenylalanine. So like that.